Okay. Hello, everyone.
Hi, Tangled.
Thank you.
Hello, everyone.
Thank you for coming. So we're just gonna wait for like a minute or two, and then we can start.
Hello, thanks God.
Hi, Camus. Yeah, so I think maybe one more minute, then we start.
Hey, Oliver. See you here.
Hi, Professor Happi.
Hey, how are you?
I'm fine, thanks. Good to see you.
Good.
Oh, Happy it's virtual, right?
Yes, Dr. Happi. I'm also here.
Hey, Oliver. I haven't seen you in years. What's going on? You are locked down in South Africa, huh?
Pardon?
I said I haven't seen you in years.
Oh, yes, unfortunately, yeah. Traveling wasn't easy.
I know.
But I'm sure we see you in next year. I'm planning to come to Nigeria.
Oh, that's good. Good.
Okay.
Let your team let me know so that we can connect when you come.
Potentially in January.
Okay.
Yeah.
Good.
All right. Thank you, everyone, for coming to this month's monthly meeting. So for those of us joining us for the very first time, this is the monthly meeting of the African BioGenome Project, and mainly looking at sequencing genomes across the continent, focusing on biodiversity. And we have a list of agenda, but we'll start with our guest speaker, Professor Christian Happi. And he is the director of the African Center of Excellence for Genomics of Infectious Disease. And he's going to be talking to us about the fantastic work he's been doing on the continent. And I must say, Professor Happi, you are really pleased to have you here. So yeah, you have the next 1 hour, basically, because I understand you need to go and give another presentation. So over to you. Basically, we'll just have your presentation, and then there will be about 10 to 15, 20 minutes question and answer from the community.
Thank you very much. I will try to run through my presentation as much as I can. So it's pretty early for me, early morning for me. I'm in Los Angeles, and then the annual Society of the American annual meeting of the American Society of Human Genetics is on. So, and I'm the guest speaker for the annual meeting. So I have to be there earlier on to get ready and then to give the talk. So I will kind of share with you guys, share my screen, and then quickly go through and see how best we can, yeah, let me just do this. Sure. I'm sure this center probably need no introduction. So, but at least I'll be talking about genomic characterization and surveillance of microbial threats. So this is a work in progress and work I've been doing over the years. And that's why you will see in this title in many places, but definitely it's not the same presentation. It's basically that we just have too much data that coming out of this program, and then we just keep showing and then demonstrating the power of genomics, and especially when it comes to genomics, characterization or genomic surveillance. I lead the African Center of Excellence for Genomics of Infectious Diseases, the World Bank, NIH, and then now a very large number of philanthropists funded, center. It is one of the two reference centers, WHO, Africa, CDC, and then World Bank reference centers in in Africa for, infectious disease innovation and especially genomic research. So, and then I also, full disclosure, you see the Sentinel here. This is our early warning system for pandemic preemption or response. So full disclosure, so this platform, we started it before the COVID-19 pandemic, and it has demonstrated to be very useful and also be the most, one of the most innovative platform, for instance, for epidemic response in the world. So, and see, okay, so this is going to be my outline. I'll quickly run through this. And quickly, I just want to mention the fact that the genomic sequencing technologies, have revolutionized the way, research and healthcare is being performed in the world. Today, we hear about personalized medicine. We hear about, gene editing technologies to actually, to change phenotypes. And all of these advances, in the field of genomics has really brought a lot of technological innovation, health benefit and economic benefit, across the globe. And while that has happened, that also created a huge disparity and a huge divide between Africa and then the rest of the world, or the rest of the world and Africa. And the reason why Africa is really not participating in the genomic revolution is simply because earlier on, a few years ago, we realized that there was really no know-how in order to participate, and there also no infrastructure on the ground to facilitate the participation. So, and in doing that, really, we felt that, in order to reduce the gap, and then and we also see genomic as a leveler or an equalizer, that Africa should really tap on this revolution in order to actually be to the fore in some areas that are very peculiar to the continent. As part of African biodiversity, so we really just decided to tap on what we call, the infectious disease genomics. If you look at this map over here, as you see it, these are the diseases or pathogens that will have pandemic potentials. And you could see the concentration of most of them actually in Africa. And that really tells you that as part of our African biodiversity, these pathogens are there. And then Africa should harness that biodiversity in order to actually occupy what I call is rightful seat in the community of nations. Africa should be the one leading research in these areas and then leading innovation in these areas. So we really just decided to focus on pathogen genomics and then genomics epidemiology as a way to really reverse, reverse the trend and then also give prominence to the continent. And in order to do that, we set up a platform, and then this platform is Africa Center of Excellence for Genomic of Infectious Disease. It is basically a platform that is really built along long-term partners between African institutions and clinical testing centers and international centers like Harvard. But this is how we started, but this has really become, the list has become extremely long, and then we have just a partnership across the globe. So our mission was really twofold. One was educational, and then the other one was research. So the educational program really was to set up a vibrant academic research environment that transcends national boundaries, where Africans could conduct relevant, responsive, and high-quality translational genomic research for development in Africa, for health development in Africa. But specifically, we really wanted to use the ACEGID platform as a way to develop a critical mass of well-trained African scientists that could leverage the knowledge and the skills of genomics and then utilize them toward, prevention and control or elimination of infectious diseases on the continent. In order to do that, we really needed to be creative, and we set up new curricula in molecular biology and genomics and bioinformatics, and then all of the curricula that we set up, master's, PhD, and then also short-term courses in molecular biology, genomics, and then and bioinformatics have international accreditation. So which means our products are pretty much equivalent to any European Union country, graduate in this field. So, but then also in order, to engage, communities in prevention effort and then health and then public health education. That is really the educational component of it that is really driven and supported by the World Bank. But, for you to do research at the level, graduate school level, they usually have a very strong, research program. And that research program that we really thought that could cut across many African countries if we wanted to do something that was intercontinental or Pan-African was really to sit down and identify the drivers of febrile illness and then therefore establishing the microbial landscape across the continent and using that microbial landscape to start thinking about developing countermeasures, such like, diagnostic vaccines and therapeutics. So that's really was really the driving force behind it. And then through the H3 Africa, and then the USNIH and many other partners, we will really set up the largest aquifever illness study, or in Africa. And really just being driven by metagenomics, by being translational about the genomic approach that we're using to this in order to develop and deliver on development of countermeasures. But in doing this, we basically was addressing one of the critical needs for genomic research and education in Africa. And that critical need has to do, is really capacity building. That is setting up genomic and teaching sequencing facilities across the continent, diagnostic core facilities, and then also enable sustainable science career among the young people that are getting trained. So I can say proudly here that, in doing this, we've been able to set up sequencing facilities across many African countries, in Cameroon, in Nigeria, in Senegal, in Sierra Leone, in Liberia. So, and then we are having more countries that we want to set up facilities, set up genomic sequencing facilities. So ultimately, our goal is actually to train the next generation of African pathogen hunters in Africa, within Africa, in the dirt rolls of Africa, start catching these pathogens early, characterizing them and developing countermeasures before they infect us, to avoid the situation in which we are today, whereby we're really playing defense instead of playing offense. So, and one of the major, the first thing that we did was really to actually demonstrate, the power of this technology and the power of this approach, to to epidemic response. And this was in 2014, for instance, when we had the Ebola outbreak in West Africa. So for the first time, during that outbreak, we used genomic as a way to guide public health response, but also to be also translational about it to develop novel countermeasures. And then when the Ebola outbreak started in 2013 in the remote confinement of Guinea and the world was looking away, I gathered a few friends, and then because I had a premonition that disease was going to spread and eventually, create a lot of casualty. So we identified diagnostics as one of the weakest link of the chain, in terms of disease surveillance. And then we went on to create the first molecular diagnosis, or we create some molecular diagnostics, train people in Sierra Leone because Sierra Leone share borders with Guinea, and then also in Liberia. And then we also train people in Nigeria because Nigeria really being the most populous country in Africa. We had a premonition that if Ebola actually reached Nigeria and then spread out of Nigeria to the rest of Africa, we would have been in big trouble. So that really helped in the sense, because this is a very first demonstration when we train people the first alert that we got in Sierra Leone, within the first week, we detected 35 cases, suspected cases, confirmed 14, and then also use our contact tracing, to nip down to the index case to a traditional heater, from which the disease spread across the country. But then this really was very clear that our approach to disease epidemiology could be very important. But beyond this, we did something that were very unprecedented in the field or in the landscape of infectious disease epidemiology. For the first time, we went on, within the first 3 weeks of the outbreak in Sierra Leone, sequence the first 99 full genome of the Ebola virus, but made this available to the international community in an open data, access format. And then also publish the SOP, that we used to do this genome sequence to enable other people to replicate what we've done. And this really formed the foundation of what you see today that have been used to respond to COVID and then to the epidemics. So in real sense, we pioneered open data sharing, we pioneered open data access, we pioneered genomic epidemiology to respond to an epidemic. And we're very glad that that pioneer work that we did in 2014 is what is being used today to respond to this global pandemic. And then really what we did was unprecedented, from data collection or for sample collection to depositing the data in GenBank. That was 10 days. That is just super quick. And then we could we were so open and so driven by just making impact in the field and using the data to guide the epidemic that we did not publish our paper until 9 months or 8 months after we submitted the data and people were using them out there in GenBank. But then what is very important and very innovative and creative about our work was the fact that, we're able to demonstrate that in the case of Sierra Leone, we have 4 different variants of the virus that came into the country. But then we were able to show that, as you track the virus over time, we're able to show that one particular variant, which is called SL3, by June 16, has taken over and became the variant that was transmitting, across the country. And then the question was, why was this variant taking over? And what was the variant transmitting better than others? So And what we did was basically to look at the genome and then study it very carefully, and also do some functional analysis later on. And we realized that there were just accumulation of mutation across the genome, among which 21 of those mutations were really, really found to be associated with increased transmissibility. And that was the very first time in the history of genomics epidemiology, in the history of an epidemic, where we could use genomic data and science to demonstrate that, a virus, because it has accumulated mutation, has acquired some additional properties, and then actually he's transmitting better and has become more than what you see. So it is the same application, or it is the same background, it is the same approach that has been used today to identify all the variants of concern and all the variants of interest. We basically are the fathers of these novel ways of doing epidemiology. The other thing that we did was basically to see what as you see here, within space and time, use genomic sequence data and then look transmission chain to establish, transmission chain within counties, within towns, within cities, within villages, and then within countries, and even across countries. So this really was the very first time you could see this kind of genomic in action, and then sharing this information with public health authorities, and then they use it effectively to respond to the outbreak, even though it was very political. What was very inspiring at the time is that as we are doing this work, organization like the World Health Organization, we're 50 years or 100 years behind our thinking. So they were telling us that, oh, no, you're doing research during an outbreak. Because, the WHO thinking at the time, was, in the previous outbreak that ever happened in Africa, they will come collect the sample, or they will send some expert in quote. In African countries, the goal was to actually come and tell Africans what to do, even though they didn't understand very well what was going on the continent. Eventually take the sample in some lab in Europe or in America, and then tell you a few years later that, oh, what was responsible for this outbreak was this particular virus. But we were doing this in real time, and then that for them was completely, they were completely shattered. They didn't understand. But through this, we were able to understand the viral factors that were really driving the epidemic. But the other thing that we did that was really unique and unprecedented was the fact that we were translational about our genomic research approach. So we went from genome sequence data and translated them into, using also genomic immunology as a way to actually translate those genome sequence data into developing point of care, diagnostics. And therefore we generated the very first rapid Ebola diagnostic that was approved, by the WHO and then the US FDA on the emergency use authorization. So the introduction of this test into West Africa at the time really became a major turning point to start, flattening the curve of the epidemic, because within 10 minutes, clinicians or doctors and lab scientists could actually have the result of a test instead of waiting for days and then keep fueling the outbreak. So this is like a church in Nigeria, for instance. This is a Redeemed Christian Church of God. And here my lab at the time wasn't too far away from this place. We were probably just about a minute, from this place. And then this is like a Friday gathering where you got about up to 5 million people gathering in a place. And this kind of thing are the things that really frightened the hell out of me, because at the time, when I look at this kind of crowd, I was just asking myself a question. So what would happen if you just have a few people within this crowd that have Ebola? This would have really spread across Nigeria and then probably across Africa as a tidal wave, and eventually, had a very major case fatality. Because we were prepared and then we knew about this, so we set up an disease intelligence system on the place, got all the necessary things, and eventually within days, when we had a suspected case, within 6 hours, we were able to confirm the case in Nigeria, made information available to public health authorities, and then work with them very closely. While we were doing testing, we were also moving on with sequencing and then using that information to actually demonstrate the hotspots and then the sinks. And Nigeria came out of this pandemic, come out of that epidemic as a case, because a country of 230 million people, where the virus appeared in Lagos, a city of 25 million people, were able to contain this outbreak within 93 days, with only 20 cases, 8 deaths, and 12 survivors. This really, went in the history and the annals of public health, because, the question was, what did you guys do? That was, so spectacular to contain an outbreak of that magnitude just within 93 days. It was really, genomic technology in action and then working with public health authorities. You could see here contact tracing and then genetic data, and then you could superimpose them, because we're using them as a way to guide. The next thing I'll talk about here is Lassa fever, which is a disease that was discovered in Nigeria probably 50-something years ago, but then I think as of 2007, there was no molecular diagnosis in Nigeria. And then, all samples in those days used to be sent to Germany, and then 90% of people would die when they are infected. This virus, is definitely a single-stranded RNA molecule, is very prevalent in West Africa, but then even serological studies will show that it's very spread, across Central Africa as well. So what we did at the time was really to a disease that was neglected. So we really focus on it and then to identify and to understand the reason why a disease of that nature could be neglected and killing people in Nigeria. In 2007, I paid a trip to do small village coming from Harvard. I paid a trip to a small place in Nigeria called Irua Specialty Teaching Hospital. What I found was a bush like this one. There was nothing right there. And then I understood that people were dying every year. They were losing about 3 medical doctors and nurses, about 5 medical personnel every year. So we went from there to set up a small rural laboratory, as you see here, and then a few months later, donated all this equipment and this molecular equipment, came back and then trained people. Eventually we set up the first molecular diagnostics on this date of September 27 in 2008, helped them to set up like a ward for clinical management. But then this is a lab that we set up right there now to do work. So you can see the evolution. But along the line, we also build capacity and then train them, and then this very remote place and this very bush has become one of the West African centers of excellence for clinical management and research on Lassa fever. But then we didn't stop there. What we did was basically to use work in collaboration with them, obtain the samples, and then eventually perform the first next generation sequencing of the virus and establish the very first genome of this virus called Lassa fever virus. And what was interesting about that is the fact that not only we established the genome using unbiased sequencing, but we're able to establish some very core fact. One is the fact that this virus emerged from present-day Nigeria about 1,185 years ago, was circulating within Nigeria for over 600 years, and moved out of Nigeria about 400 years, westward. And then you will see the implication of this, toward the end of my talk, which is basically that we established not only that in Nigeria, we have different variants circulating, 3 in the north, 2 in the south and south, and then and 1 in the northeast. That for us was very important because not only we use all of this information to just understand the epidemiology, but we also translated them in developing, what I call a pan-Lassa, because having all these different clades of variant, pan-Lassa rapid diagnostic tests that could pick up all of this, within a short time. So that really what became very important. So we had this published and then generated all this and then have this in Cell, and Cell was able to give us a cover. But more importantly is the fact that in 2018, Nigeria was facing a surge in Lassa fever. So in 8 weeks, the country faced more cases in 8 weeks than in the past 2 years combined. There were many public health questions that were asked. Were we dealing with new variant or more virulent strain? Because the case fatality was also higher. So was this surge due to human-to-human transmission, or that the diagnosis were not correct? And what we did was basically to swing into action and use real-time, genomic to help the response on the country. Collected sample across the areas, did rapid sequencing, and then provided the response. We sequenced 300 genomes and then used the data to explain to people, to public health authority what was happening. And the answers were very clear. We didn't find any evidence of a particular new variant strain, no evidence of sustained human-to-human transmission, but what we found was that the surge was due to multiple introduction of genetically independent, viruses of known lineages that we know 1, 2, and 3, but also we demonstrated that was coming from animal reservoirs. And then we also showed that lineage 2 that we showed before has split into 2A in the southwest and then 2B in the southeast part of Nigeria. But what is interesting, if you look at all of those lineages, they are separated by major a natural geographical barrier. You have the Benue River here, you have the Niger River here, and then that really tells you that the mastomeres or the rats that were contained in this could not swim across those rivers. We had it published in New England Journal at the time, but then what was important about it is the fact that we actually opened, us a new vista, which means that we started really looking into genetic diversity in rodent animal, and we're also going to establish some very clear-cut fact. This is the first time we established, getting a sample from an animal and then in a human, and we see the same virus establishing clear transmission, between the animal and then the human. So that really became important, but we really didn't stop there. What we did was basically to go beyond it and then use the data, again, leveraging gene editing technology to create a novel generation of rapid diagnostic tests, as you see here, using the CRISPR-based technology or CRISPR gene editing technology to create a very highly sensitive, diagnosis for Lassa fever, for Lassa fever. And then using machine learning, so studying the gene sequences, identifying the guide, the targeting guide, using sequences to create the guide RNAs and then see what the guide could interact with, identify sequences that were very unique to very lineage, and then use that as a way to create this diagnostic that you see here, and then at a cost that is really way cheaper than PCR, which is about $3, And then you could see that here, this is where a team of Nature came to my lab and it was embedded, and for 1 week, and it's one of my students, my former graduate student that is in University of San Francisco, California today. But then here you could see, so we were basically, and this was the very first, CRISPR-based diagnostics for an infectious disease that was ever developed. But we basically also look at not only at the sample itself, but we also look at the genome of the human genome. The reason why we targeted the human genome was simply because we noticed that in the communities where we were working, some people get exposed to Lassa, but they don't get sick, while other people get moderately sick and other people get severely sick. So the question was, our assumption was that it's most likely due to some genetic selective pressure, or some natural selection in population. And some part of Nigeria, for instance, in the southwestern part of Nigeria, you rarely hear about Lassa fever, even though people get exposed to it. And then we went on to look at the disease, recruiting people across the Yoruba, the Ishans, and then the Mendes of Sierra Leone where we were working. And then you could see, again, here some signals of selection. You could see that very high in the Yorubas, in the Ishans in Nigeria is a bit higher, but very, very low in the Mendes of Sierra Leone. And this really is very much associated that we found in the end, associated with the age of the virus, and then the fact that the virus probably emerged in the southwestern part of Nigeria about 1,000 years ago. So that gave you that kind of high level of frequency in some part, depending on where you are, doing genome-wide association studies. So we're able to find out some very interesting thing, which really is the fact that, we're able to see the virus really spreading out of Nigeria to the rest of the world. The Yorubas, for instance, have a very, very high level of selection. But what is important is the fact that, we identified this in one of the genes, in the human gene called large. And then the large really, we realize that, it is a gene that really is very important, and then that really is involved into entry. That it expresses a protein which is called the large proteins, and that is involved into the entry of the cell addition and then penetration of the virus into the cell. So we realize that, depending on that, for alpha-dystroglycan, which is a protein that is involved in cell addition on the extracellular membrane to function optimally, and then to bring in the cell into the virus into the cell, it needs to be modified by large. So, and then we also, by previous work done by Stephen Coons of late memory, I realized, we found that post-translational modification of alpha-dystroglycan could be involved is highly involved into it. So we found that, alpha-dystroglycan is a putative, receptors of all arenavirus, and Lassa virus is one of them. So, and then we think that, we went on to because of the mutation, and then did some functional analysis, and we realized that the signal at large is involved, but then the mutation actually within that signal is actually involved also in blocking the entrance of the virus into the cell. So we basically feel that, as a dystroglycan is modified by large, as you see here, that's a large protein, and then that alpha-dystroglycan modifier goes into the surface of the cell. When the large is not mutated, for instance, it facilitates the penetration of the cell, entry of the cell into the viral particles into the cell. But when large is basically modified, then it actually blocks, the entrance of the virus into the cell. And then we have functional data now to demonstrate this, and we have a paper coming out in Nature, to do this work. But I just want to let you know, this is a 10 years project that we've been running, but eventually we just got the hit recently, and Nature picked that up, is publishing it. But then during this work, we also really did something that was unprecedented, that is basically conducting the largest clinical metagenomic studies in Africa, because we were dealing with clinicians that were telling us that people present with different symptoms of viral hemorrhagic fevers or fevers, and they all appear negative, but then when they treat with some medicine, then it works. So we really just went on to do a case control study, basically taking people with febrile illness and those without febrile illness, with the idea of just doing metagenomics to identify the different pathogens and then leveraging them that as a way to develop the necessary countermeasures. So conducting this study, we really stumbled on something that was very important here. So in the process, we discovered two novel rhabdoviruses, and we call those rhabdoviruses Epomavirus 1 and Epomavirus 2 because they were discovered there. And then that's really one of the two major discoveries that were done in the field of virology in the past 10 years. These two novel rhabdoviruses have been associated or had very close similarity with another rhabdovirus called the Bakongo virus in the DRC that was involved in the viral hemorrhagic fever in the DRC in 2012. But in the case of Nigeria, we didn't see that. And then when we used the sequence data to create, an ELISA test to actually to just look at exposure. By the time we went into the communities, we realized that about 80% of those people were involved had exposure to the virus, and that really buttresses the point that we have a lot of pathogens that are circulating that we don't know much about. And then we should rather be talking about emerging diagnostic instead of talking about emerging or re-emerging disease. This is another use case where in Nigeria, in November 2018, within a week, within 2 weeks, Nigeria lost about 179 school children. All of the testing, malaria, Lassa fever, yellow fever, and many others actually proved to be negative. So the Nigeria Center for Disease Control sent 22 samples in our lab. We went on to do microbial metagenomics using, advanced sequencing platform. At the time we used the IC4000, and we were able to establish something very clear, that the infection and the death of the children were actually done, due to yellow fever. But what was peculiar about the yellow fever strain that we discovered in those children or in that environment was that they were very different from the ones that have been circulating in Nigeria. So you can see this is a cluster here, and then these are the ones that have been circulating in Nigeria before. So they were very different from the ones that have been circulating in Nigeria in the past 94 years. And this information was shared immediately to the Nigeria Center for Disease Control. They declared an outbreak, and they were able to contain that outbreak within 2 weeks, saving thousands of lives. This is another use case, for instance, where you see in Lagos, within the same period, a parent bring their children, one of the children to the hospital. The child was bleeding from all orifices, nose, ears, mouth, and everything. And then the pediatric ward within seconds, got empty because everybody was running for their life. They thought Ebola was back in the hospital. We deployed our team down there, collected the sample within 72 hours. We established that viral hemorrhage or that hemorrhage in that child was due to enterovirus B. So this information is passed back to the hospital, the confidence is built in the medical team, and then they resume work. So this is another use case whereby we have a situation whereby people, we have patients within Lagos that are not responding to treatment, and then they are given all kinds of antibiotics. And of course, they were demonstrated to have, these are TB patients that are not responding to patient. Gene expert, they must show that, these patients have MDR, but then that really does not resolve the issue, because when the samples are sent to us, we actually use metagenomics or bacterial genomics actually to show clearly that it is extensively drug-resistant tuberculosis and not MDR, basically showing the relevance of this technology and how this should be embedded into diagnosis, because this was the first report of extensive drug-resistant tuberculosis in West Africa, in Nigeria and West Africa. Then we're not just doing this on humans. So this is, again, a case where we basically using a One Health approach. And then we had a rabbit hemorrhagic disease in Nigeria. This is called the Ebola of rabbits, because the animal bleed from all orifices, and then eventually the case fatality about 90%, if not 100%. So when this happened, for instance, sample was sent down to a lab. We got the sample and then went on to sequence it. And then we actually found that it was the rabbit hemorrhagic disease virus, and then the lineage 2 or clade 2 of this virus. And this is the very first time, this was characterized in sub-Saharan Africa. And this disease is very dreadful, because when the U.S. Department of Agriculture heard about it, they reached out to Africa CDC. They wanted the sample to be shipped to the U.S., but unfortunately, Africa CDC told them that, if it is in Nigeria, we know that lab definitely will have sequences for you and our samples. So basically turning the tide to let people know that, the time for shipping sample is over. So I think we have the ability to create data in very quick time. So we do this for mosquitoes as well. So determining, using metagenomics as a way to understand what is circulating in mosquitoes. This is during a yellow fever outbreak in Nigeria. But apart from yellow fever virus, we also identify an incredible number of viruses being carried by mosquitoes. And that becomes very relevant in the field of this. So what do we do with all of this information? Actually, we use all of this information to set up, Sentinel. Sentinel is a pandemic preemption and response system that we set up at the time in 2019 before this outbreak, and it's built on three pillars. That is early detection using all of this technology that I showed you, creating CRISPR-based diagnostics that are deployed in the field. There are point-of-care diagnostics that can detect, any pathogen within 5 minutes or within minutes. Or if the test is negative, then we also have a couple of tests. Combine all of those tests into a multiplex platform that can detect up to 469 pathogens in one single assay. And then if it's negative, you went to the sequencing hub, and then you can discover new viruses, create, a CRISPR diagnosis within a week, and then that can go back to the field. So while those testings are done, the reports are also being given in real time. So thereby we create dashboards, and the dashboards provide the information to policymakers either on your dashboard or on your phone, and then you know exactly where a test has been done with the GPS with all of the information. This really empowers people to actually do what I call coordinated and targeted response, because we know that in order to contain an outbreak or an epidemic, you just need two things: speed and accuracy. And that's what we basically work on. This is basically that I explained. You have this thing at the remote clinic, and then we've really turned this into something that is very transformative and impactful. So imagine, what we are basically telling you here is that you are doing PCR on a sheet of paper. You really do not need any equipment free. You basically have everything on a sheet of paper. You just stuck this in your mouth. You can get saliva, take it out, and then the reaction takes place there, and then you can have the result. The higher the temperature, the better. So that's really the thing. And then you have a clinic in the hospitals, nearby hospitals. We definitely have established what I call MCARMEN platform. I'm going to talk to you about it, where we couple and combine together 469 different pathogens that we know well characterized globally. And then you could actually just do that in one single sample, but you can also multiplex that in plates of 96, plate of 384, and even more. Then you also send the sample to the Genome Sequencing Center, where you can actually just better characterize and use that as a way to refine your test. And then all this information, as we say, at each level are transmitted in real time, in public health to public health networks, so that they can use it to respond. So basically, it's built on the hub and spoke, model there, whereby you have the genomic hub, and then you have the spokes around it, and then you also have them testing in the remote areas. So, and then, the connection is basically where you really just transfer information very quickly, so that public health information will know. But then the third point is called empowered. So where you're empowering frontline healthcare workers in order to use this genomic and then all these information technologies, tools, in order to detect early and then pass information in real time. So, and ultimately, our goal is basically to use all of these actually to create what I call going from the state-of-the-art technology, bringing it down to point of need or point of care diagnostics. I use that as a way to expand the scientific capacity, to Africa in order to ensure independent surveillance and outbreak response. So, and this basically came into play when we had COVID. Remember that COVID started, first of all, in the case of Africa, in Egypt. And then from after Egypt, it was South Africa. But Nigeria was the third country actually to have been affected by COVID. But then what we did was really with COVID, we actually put our big guns. We had our next generation sequencer, the NovaSeq, which is the most performing sequencing platform, even though we also got a Sequel 2E from Oliver. But we have all of those high-performing, platforms. What we did was basically for COVID, when the first suspected case happened in Nigeria, within 48 hours, we had already sequenced and assembled the full genome, then put all of the report, share the report with the Nigeria Center for Disease Control to show that this wasn't a local case, but it was an imported case. And that really resulted, in the first SARS-CoV-2 genome sequence in Nigeria and Africa. So you could really see the firepower of this technology because, leveraging this, even though Nigeria was the third country to have COVID, and weeks after Egypt and South Africa, we were the very first to actually do the first genome of COVID in the speed of light, 48 hours. So that really showing the firepower of this, but then over the time, we work with Nigerian public health authorities, as you see here, for instance, over space and time, create dashboard like this one, knowing exactly in real time what was circulating where in Nigeria and use the information to guide public health response. In this, for instance, at some point, we knew that in Nigeria, we had 49 different variants that were circulating and public health authorities were using them actually as a way to respond because there was no way in Nigeria at each point in time, the presidential steering committee would take a meeting without us giving them the report. So they were guided, but we also use this as a way to really train other African countries how to do it. This is the case of Nigeria, for instance, where you see this over space and time, the different variant that came into the country, but we also leverage, our ability, to create novel technologies or to translate technology to create what we call another CRISPR point of need or point of care diagnostics. These are the very first one that we did in our lab. You could see the raw result here, but then this has really been out now and has been used extremely well. So using all of the data that we've created and all of the genome sequence that we've done for yellow fever and everything that we've done together, we've now created what we call MCARMEN, which is a combinatorial arrays of multiple CRISPR tests, and then really doing this in massively parallel analysis that can enable you to do one single test, and then you can just create this. But we've also trained a lot of people in the period, and then within the time, we've trained over 1,300 people across Africa. You can see my team here in the midst of the pandemic, moving from one lab to the other, training people how to do some of these tests. And then in collaboration with the Africa PGI, for instance, you could see this map. We're very proud of this map because, within 2 years or within the past few years, we've been able to train 42 out of 54 African countries. That really more than 2/3 of the African continent. And then really that has resulted in terms of increasing the number of genome sequences that emanated from Africa. So, and then this has resulted, as you see here, at the end of 2020, Africa had only about 5,000 genomes, but by August, we had over 60,000 genomes because people were trained and then they were going back to do it. And then toward the end of the year, we'll pass over 120,000 genomes. So really showing the impact of these trainings and the impact of sharing these technologies across the world. So we use that as a way to work with other partners to identify variants of concern, variants of interest across the continent and show you how this moves across the continent. But I'm telling you about developing this dashboard. This dashboard become very important because this is one of the dashboards where you look, it gives you all of the variants. It shows you how this vary over time, depending on where you are in Nigeria. You could look at between those people that are vaccinated, the frequency of this variant on unvaccinated. You could look at the different places where you have this in Nigeria. You could look at the trend over time, different depending on where you are. So really having this, you don't have to be a genomist because there's a lot of back-end analysis, but you just see the visualization. I really want to say that what we've done also is actually to work on monkeypox and then leverage our knowledge and the skill and the voice that we have, in order to tell organizations like WHO that they are not responsible. But unfortunately, through telling them that they are irresponsible, they've been able to come to some good sense, and then we're able to recently call on the change of the name of monkeypox because we told them calling all these variants, West African variant, South African variant, or whatever they call them, is unhelpful because we had a variant of monkeypox that emerged from Europe, but then there was no nomenclature to call it something within Europe. So, and then we challenged them, and then they agreed that they will need to come to a more non-discriminatory and non-stigmatizing nomenclature of monkeypox. Really, at this point, I really want to, thank again for the opportunity, but then this is always in the name of some of our colleagues that die along the line, working, on this very dreadful disease, and they really remain a source of inspiration to us. They didn't just die in vain. We'll keep pushing in order to really make sure that Africa deserves its the rightful place in the Committee of Nations. So I really want to thank my team for all of the effort that they're doing. I'm basically speaking on their behalf, and then also acknowledge all of those that are funding us, and then and then also thank you again for the opportunity. Over.
Oh, thank you, Professor Happi. That was quite a lot of things, basically. And thank you for speaking to us today. So we're going to very quickly take some questions. So I understand you have to go back to the live keynote in LA. So just very quickly, we'll just take some questions from people. Does anyone have any question they want to ask Kristen Happi? You can just raise up your hand and I would. So DNALC, could you please unmute and introduce yourself, where you're speaking from, and yeah. DNALC in Nigeria.
Okay. Can you hear me?
Yes, please, go ahead.
Okay, so good evening or good morning, Professor Happi. Good morning, thank God. So thank you, Professor Happi, for this enlightening lecture. I took your course first at Abonlao Lab in 2015, and since then I've followed you. So right now we are running a DNA Learning Center here at Enugu in the Southeast. But then the question is, what can we do to partner with you to have more of scientists who come from the Southeast be trained to get our own genomic data worked on? Because there's a lot of work that needs to be done from this part of Nigeria that is not being done. And then we have the resources, we have the center, we have equipment, but then how do we partner with you to get more work done from this side? I don't think more has been done on the Southeastern genes and diseases. So I think we're willing to take that challenge, but we don't know how we can partner and then get more of our people trained to begin to do this work.
Yeah, we do have a research program in the eastern part of Nigeria. We have just built a new lab in Abakaliki Hospital, and we do have a network of laboratories. And then one of the things that we're doing basically there is actually we're building some what I call small rural clinics in many places across, and we're definitely doing that. I mean, for training, I don't think that is a problem. We have what I call a genomic boot camp that will train people across Africa. So I think it is a program that is highly competitive, and then I can only tell the people that are interested, to apply, and then eventually, people can use that as a way to attend the training program. So if we've trained people across Nigeria, I know in Nigeria is actually the country where we train most of the people. So in this training program, but it's all about people showing interest and then applying for the program. I know in the past 2 years, we haven't applied, we haven't done the program because of COVID. We got too busy with COVID work, but we're hoping in 2023, we should be able to start a program. So I think it should be open. You check the website of ACEGID, and then people from the eastern part of Nigeria should apply.
Okay, thank you.
Thank you, Professor Happi. And yeah, okay, over to you, Eric, please.
Yes, great work that you're doing and very comprehensive. So I wondered about the large gene area that you talked about that was showing, selection in Yoruba people. And I'm wondering, was this selection for resistance against the virus or for more susceptibility to it?
Selection for resistance is on chromosome 22. So you're going to see that popping up in nature very soon. So it's really, I mean, it's 10 years work with sequence over 3,000 genomes, to do GWAS and all of that, to get that signal. And we've also seen some selection in the Ishans people, but it's not as strong as the Yorubas that we've seen. That is also has to do with the age of the virus. But you also notice that over time, when you look, I think I was telling people, and one of the things I'm going to be telling them that as a keynote speaker for the American Society of Human Genetics this year, I think a lot of the genetic variation that you're seeing in African population or people of African ancestries are probably driven by a lot of these infectious pathogens. Because of this selection, you're going to be seeing those variations, and that becomes very critical, and because you're having that, and then because of all of those things will come at fitness costs, you're probably going to be having other mutations accumulating in other parts of the genomes, and then that results in some of those other things that you see, cardiovascular diseases and then renal diseases. You think about the selection of APO1, for instance, in African population due to African trypanosomiasis. I think this is the kind of dynamic that we're seeing right now, where you see a lot of signal. Think about malaria and then sickle cell. So this is kind of stuff that we're seeing, and then I think for me to, identify this signal in Yoruba people really is very significant because again, one of those first, discoveries that we're basically just having a clear example to show, that you have those mutations that make some people be resistant. And for people that know Nigeria, you would realize that anytime you have this Lassa fever outbreak, you rarely hear about it, in the part of the Yorubas. And that my experience over the years has really demonstrated that, I kept asking why. The few cases that you see, you realize are imported cases. And I think this time along, we got the answer, and then really is beyond, beyond any doubt.
And it's a very large genomic region, it looked like. So it's more than one gene, I take it. Or that gene is very large. The large gene is very large.
It's actually the largest gene of the human genome,
Okay.
but we really narrow it down to one specific place. So we have it. So it's on chromosome 22. So we really have that signal, and it's very consistent.
All right, good, thank you.
Thank you, Eric. Over to you, Kellen, please. Kellen Ephraim.
Good evening, everyone. I'm Dr. Kellen Ephraim. I have a question, sir. I'm from the northern Nigeria, and we realize here in the north, we are left behind when it comes to research, most especially research of this nature. I carry out a research during my PhD, and I realized in the whole of the north, that was the first research to be done on snail. And you know how the north is, we are highly populated, and any outbreak, it gets to circulate fast as possible. I don't know if there is a way this particular program, we can be trained into the program. Please, sir, I don't know if there is any way they can help the northern part of Nigeria so that we'll be included in this program. Thank you.
Our training program is very open, and I've trained quite a number of people from the northern part of Nigeria. I've trained people from ABU, I've trained people from University of Maiduguri, from Jos. I've had people from one Dr. Kia just spent time in my lab recently with 3 of our students. She came from ABU. So I think I got quite a number of people that come to train and then learn some of the skills. I guess it's all about interest. I guess it's all about how you want to do it. And I just want to say that the training program is pretty open. It is also quite competitive. But I guess if you have a research question that you want to answer, so I think you can just check the website and then you go to info@ACEGID, and there's always somebody that will address the question. And if you say if it is anything that is of interest to us, we will do. But the training program is very open, and then watch out for the 2023 program, so training program, so that you can join. And what is actually even interesting about our program is that we are very female biased in the selection of our candidate, in the sense that because we think that we can use this program as an equalizer to reduce the gap that is existing between, especially in faculties and at high education level, in science, between male and female. So I think we basically will favor more female training than male trainings.
Thank you, Professor Happi. So I understand you need to go in a few minutes, but we have a few questions for you. So I think that the first one is, given the fact that the ACEGID laboratory is one of its kind on the continent, and of course you may have already heard about the African BioGenome project and what the project aims to do. So the idea is to leverage on existing facilities on the continent, and in order to be able to sequence things. So it would be nice to, again, maybe it may not be in this meeting, to discuss how the project could partner with your laboratory, because, for instance, of the many machines and sequencing platforms, you just mentioned the NovoSeq, and again you mentioned the SQL 2. I'm sure your laboratory is maybe one of the 2 or 3 laboratories that have those 2 machines in the same place. So it would be nice to see how we would explore those opportunities. And in terms of sequencing, you're already sequencing vertebrate like rats and others. Again, see how we could look at other aspects, like, I don't know if sequencing plants and things or eukaryotic microbes, but those are conversations we might have to have at some point to see how, from in the West African angle, to be able to leverage on those existing facilities and also the capacities you've already built. So you don't need to answer on this platform, but it's something we would have to discuss at some point. But if you want to make a comment, that would be great.
No, I just want to say that, our lab is pretty open to, it's not something that is really located or restricted to West Africa. So I think in the course of this pandemic, we supported sequencing for about 26 African countries. So I think from Somalia to Egypt to even Libya, so to countries like Tunisia. So that map that you see there, we actually supported them in sequencing many of their samples and then send them back to them. So I think it's not, we're not restricted to a particular region. But at this point, I'm not sure how much we can process in terms of other set of samples, but we can do some pretty cool human genomic work. We do some pretty cool pathogen genomic work. We haven't done orders. I know that somebody was talking to me. I think I had a discussion with Luke Man. He's telling me about discussing some plants and then some trees and all kinds of stuff. I guess it's part of this whole stuff, but I don't know. So I guess it's something that we have to see, but then we're also expanding our sequencing platform and expanding the lab. So I think we're definitely going to be having many more sequencers that are coming. We've invested in getting the NovaSeq X that just was released by Illumina. Really? I think we're going to be having 2 pieces delivered in our lab.
That's great.
And then in the next, I think it's the second quarter of 2022. So I think, and then our lab is also expanding, and we're having a huge sequencing core that may be having probably the most advanced sequencing platforms. I mean, Oliver is here, hopefully with resources, we'll probably get acquiring additional sequels. We want to have all of the platforms in the same place, and then so that we can leverage, the technologies available.
Yeah, I think perfectly. That sounds like a good opportunity to be able to coordinate on that, because that would the projects and the sequencing of the species will also maximize the usage, of the platform. But I know that AfricaBP does not aim to put so much pressure on the infrastructure, but it's something that I would like to explore. But maybe we can take it out of here and we'll be in touch. And I'm happy that Lukman has already been in touch. So you'll begin to see more of this from either Lukman or others who are based on the continent, from the NovaSeq angle and also from the SQL2 angle as well. And I'm happy that you'll be also happy to give us access to this, and we can work together, and but we'll basically have that conversation. So yeah,
thank you. I will have to leave you. I mean, already. So thanks.
All right.
And then
okay, yeah, and thank you very much. And yeah, we'll be in touch. I understand you have to leave now. All right. Okay, so I think we're gonna we have some items on the agenda, but maybe we can just quickly see where we are. So Josiah, do you want to talk about the partnership?
Yeah, I have something small to share.